This is another of our series of interviews on innovations in science, particularly medical science at Hebrew University, in particular at the Institute of Medical Research, Israel, Canada, and our guest is Abraham Fainsod. Welcome to the show.
Thank you for inviting me.
Oh, you're most welcome. Now, we'll get into your breakthroughs on fetal alcohol syndrome, but we want to build toward it. First, by starting with, how did you get into this field?
How did I get into this field? I came to Israel from Mexico.
From Mexico?
Yes.
I see.
And I did
How old were you?
18.
I see.
I did my undergraduate studies at Hebrew University. I also did my PhD in the Department of Genetics, working on cell division, regulation of cell division.
Why did you choose that field? What was the attraction?
Regulation of cell division?
Yeah.
The professor.
I see.
I really liked the professor that was working on that field. And then for advanced postdoctoral training, I had to find a niche for myself. And at the time, I was looking on genetic regulation of embryonic development. So that's how I got into the field.
Now, how's that field had a big breakthrough in the 30s with an experiment in the 20s. And then it stalled as I read the literature, and I was in medical school, and that's what seemed to be the case. Is that correct, or what's the vision of the?
It wasn't really stalled. What happened is there were no tools. I mean, there's the original experiment of Hans Spemann and his student Mangold, and that showed many basic principles in developmental biology, like a cell can instruct the neighboring cell to do different things. It wasn't known until then. There was no proof. And they could show it. And the experiment is a very simple one. They took a group of about 40, 60 cells from one embryo and transplanted it into a host embryo, and they got Siamese twins, which means that the new cells managed to organize a whole embryo around themselves by providing signals, instructing the neighboring cells. And that opened a lot of questions in biology. How does cells communicate? How do you pass information from one cell to another? There were no tools. Many people worked, they tried to do, for many years in the 40s, there were experiments done mostly in chicken embryos, actually. And in 1991, the first gene expressed in those 40 cells was identified, and that opened the way to use molecular tools.
So it's really the last 20 years it breaks open again.
Yes.
And all from that original experiment in the early 20s.
Yes.
He got a Nobel Prize in the 30s, in the 35, I think?
35.
But there must have been Nobel Prizes come out of this latest 20 years?
Actually, in developmental biology, there was in 2005, if I'm not mistaken, there were two scientists, three, sorry, one from Germany and two from the States on developmental biology, and that was the jump to genetics. They identified the genes that regulate the process.
These organizer genes.
Not only organizer, most of these people worked in flies.
I see.
But the fly genetics identified the initial genes that paved the way for.
But you work with frogs.
I work with frogs.
And why frogs?
Actually, I started with mice. Mice at the developmental stages I'm interested. The mouse embryos are tiny and very difficult to work with. And then when I established my laboratory in Israel, I moved to chicken embryos because there was a large group of chicken embryologists in Jerusalem, actually. And then I wanted to do more genetic manipulation, molecular manipulation. And so for my sabbatical, I went to learn how to work with frog embryos.
And where did you go for your sabbatical?
UCLA.
Oh, I see. So that gave you a whole new...
Yeah, a whole new experimental system, and sometimes you find the chemistry also with the experimental system, and I like it.
But it also broke open a whole new area for you working with frogs, didn't it?
Yes, we started asking different types of questions. We could manipulate gene expression, gene function, and then ask specific questions. we're interested early embryos in most organisms tend to be either a ball or a disc, like a CD. So they don't have a head or a tail. And at one point, the embryo has to define where is the head and to put the tail in the right place. And then there's other axes, the back and the belly, left, right. So what's the genetic regulation to establish that? And also, if you have the head and the tail, you also have to fill in all the information. And our neck is different from our chest. So all the genes that work in that process.
It's quite complicated, but you have to identify the specific gene and gene set to work with that have to do with a specific function, I assume.
Yes.
And it's what, multiple locations?
Also, but what happens is we don't have that many genes. Overall, we estimate that a human has about, human genome has about 28,000, 30,000 genes. So genes have to do many functions. And sometimes we use the same gene over and over again to do similar things, but in different places.
So how did you get into the alcohol issue?
The alcohol issue, we were interested in the head-tail axis. And we know that this axis is regulated by retinoic acid. It's a molecule. It's actually what we make out of vitamin A. A different name for vitamin A is retinol. It's an alcohol. And we were looking for ways of manipulating this vitamin A derivative. And a student of mine came across a model, a suggestion that perhaps alcohol might starve the embryo for vitamin A derivatives.
This was a suggestion by some other scientist?
Yeah, a scientist in California wrote a paper on a nutrition meeting for nurses. And then we started reading, and first we noticed that people haven't really used modern tools to study this problem, not in embryos. And so we continued studying, and then the student came and asked me, can I drop the other project and can I work on this alcohol question, which seems to be very interesting.
So what do? You get the frogs drunk? Or not the frogs, the embryos.
The advantage of working with an experimental system as the frogs is, number one, we have hundreds of embryos, which we can do experiments on. We can manipulate the gene function, and they grow in a petri dish in a water solution. So if we want to treat them with alcohol, we just have to add alcohol to the petri dish.
So very simple.
And that's very simple. Yeah.
And big breakthroughs come through just such simplicity.
It's simple, but I mean, the student at the time had to calibrate amounts of alcohol, what stages exactly to give it, just to make it relevant to human disease.
And the breakthrough you make, basically, I mean, it's considered, I think, from my reading of the papers, though I'm not a scientist, it was a big breakthrough in an initial really understanding the mechanics of this process that everybody had been looking for.
Yes, people have suggested several models for how the alcohol might be affecting the embryo. We actually didn't know exactly when is the high sensitivity window, which we could do in the frogs because we had hundreds of embryos that we could put in or take out of alcohol at different developmental stages. Nobody knew what structures in the embryo were being affected, and now we know that it's the Spemann organizer, this group of cells that he identified. And we also went on to try and study the biochemistry, exactly how is the alcohol affecting and exactly which protein it's competing with, and starving the embryo actually for retinoic acid for this.
Well, we'll go into the whole nature of fetal alcohol syndrome, but we'll take a break first and discuss that, and then go back to the science, because I'm fascinated by the way it works and how the distortions it creates. But we'll take a break first.